O-Level Biology 5090, Chapter 13, Excretion. Excretion is the removal of metabolic waste out of the body. The examples of excretion are: number one, carbon dioxide is a waste product of respiration. It is excreted through the lungs. And the other example of excretion is the production of urea, which is a toxic waste product, and it is produced in the liver from the breakdown of excess amino acids. The next, the human excretory system. And the human excretory system consists of many parts. Two kidneys are there, which are bean shaped, and blood is entered into the kidneys through renal artery, and renal veins take out the blood from the kidneys. From the pelvis side of the kidneys, two ureters arise, which open into the bladder because the ureters transport the blood from the kidney towards the bladder. The urine is stored in the bladder for some time and it is excreted through urethra. Now, I would like to say about urea, which is a nitrogenous waste, toxic but less toxic than ammonia. It is excreted through urine. If its concentration is increased, so its toxicity is also increased. So our kidney and urinary system help the body to eliminate liquid waste called urea and to keep chemicals such as potassium and sodium and waste water in balance. Next, the unit of kidney is called as nephron. So in the first slide, we will discuss the parts of nephron. Nephron consists of a Bowman capsule, which is cup shaped, and after the name of the scientist, which his name was Bowman, it includes a tuft of cilia or a filter, which is called as glomerulus. Blood is entered into the Bowman capsule through renal artery, which carries the oxygenated blood, and it leaves through renal vein, which carries oxygen depleted blood. When blood enters into the glomerulus, it is filtered over here, which is called as ultrafiltration. It is called ultrafiltration. And after ultrafiltration, the blood enters into the second part of the nephron, which is called as proximal convoluted tubule, then loop of Henle, and from loop of Henle, the blood enters into the distal convoluted tubule, and from distal convoluted tubule, collecting tubules or collecting ducts are there. So these are the different parts of nephron. Now in the next slide, we will discuss the function of the nephron. Already told you that the two major functions takes place in the nephron. They are ultrafiltration and selective reabsorption. Number one, ultrafiltration, which takes place in the Bowman capsule through the filter, which is glomerulus. So ultrafiltration takes place here in the glomerulus when blood enters through great pressure. Then ultra or fine filtration takes place here. Then the filtrate enters into the second part of the nephron, which is proximal convoluted tubule. Then the selective reabsorption starts from the second part. So the reabsorption of ions, water, nutrients takes place, but the toxins are removed into the filtrate to adjust the pH. And the other part is ascending and descending loop of Fanley. In the ascending loop of Fanley, the reabsorption of sodium and chloride ions takes place. And in the descending loop of Fanley, the reabsorption of water takes place. That is, the water enters into the interstitial fluid from the filtrate through the aquaporins, which are the small pores present in the walls of the nephron. So the collecting tubules are there. Before collecting tubules, we have distal tubule, which selectively secretes and reabsorbs different ions to maintain blood pH and electrolyte balance. So electrolytes balance is caused as they are reabsorbed, pH is maintained, and then reabsorption of solutes and water from the filtrates will take place in the collecting tube. So in this way, these two major functions takes place. In the first part, ultrafiltration, and then the selective reabsorption of the different substance. In the next slide, we will discuss the composition of the filtrate and ultrafiltration and selective reabsorption of these different substance from the filtrate. Blood enters into containing metabolic waste products into the Bowman capsule, and the ultrafiltration takes place in the glomerulus, and formation of filtrate takes place, which enters into the second part of the nephron. In the filtrate, amino acids, glucose, salts, water, and urea are present. Amino acids, glucose, salt, water, very important components which should not be excreted out. So, in the second part, selective reabsorption takes place, and due to selective reabsorption, amino acid, glucose, salt, water are reabsorbed, and then excess water, excess mineral salts, and urea removed through urine. If water is not in excess, it will be reabsorbed. If it is in excess, it will be excreted out. It will be removed. So the urea and the other wastewaters will be removed out of the body. So in this way, in the nephron, these two major functions of ultrafiltration and selective reabsorption takes place. Assimilation of amino acids. First of all, what are amino acids? Amino acids are the fundamental building blocks of protein, critical for numerous physiological processes. These compounds are derived from dietary proteins and absorbed into the bloodstream through digestion. So amino acids are the unit of proteins and their first role in assimilation. Post digestion, amino acids are transported to the liver. After absorption, they are transported towards the liver, and the liver acts as a regulator, maintaining amino acid levels in the blood. It synthesizes new proteins from these amino acids for use in various body functions. So, protein is synthesized in the body. So, in the next slide, we will discuss how this protein synthesis takes place. The process of protein synthesis is protein synthesis involves linking amino acids in specific sequence and dictated by cellular genetic information. Amino acids will join together to make a chain of amino acid. They will join with the help of peptide bonds, but the sequence of these amino acids in the formation of a protein is dictated by a cellular genetic information. The process takes place in two steps: transcription and translation. So this process is crucial for producing new proteins that the body requires for different functions. So protein synthesis is very important process. which takes place in the body. The next slide is the deamination. Deamination is the removal of amino group from the amino acid. When the amino acid is changed into ammonia, the enzymes that stimulates this action is called deaminases. Deamination principally occur in the liver. So this is the actually the function of the liver, deamination, in which amino acids are broken down by removing the amino group. However, glutamate is also deaminated in the kidney. Glutamate is another amino acid which is deaminated in the kidney. So dissimilation of amino acids generates nitrogen containing amino acid NH2, which are harmful to the cells. Through deamination, the liver alters these products into non-toxic ones. The amino group is first changed into ammonia. It is very toxic. Subsequently, the ammonia is changed into the urea, which is dispersed through urine, but urea is less toxic than ammonia. So in this way, deamination will take place. Thanks for watching my video. It is all about the excretion according to the syllabus. Please like, subscribe, and share my video